So I'm going to give a brief introduction and then turn it over very shortly to Jay, who has a massive amount of data to present. We first want to express our deep gratitude to Paul Allen and the Foundation and to Kathy and Tom for their support organizing this meeting and making this all possible. So the C. elegans fate map is a roadmap to development that in part was responsible for Sulston and colleagues' 2005 Nobel Prize. It was constructed very laboriously by sitting at a microscope and actually recording the birth of all cells during worm development over the better part of a day. And when you can look at an exploded view, it actually gives you the precise lineage history of all the developed cells in the worm. And while we possess this very fundamental milestone that has contributed enormously to developmental biology for the worm, we're lacking it for other model organisms, and in particular, Homo sapiens. And it's not so easy to do in other living things, and that's because we are dealing not with 1,000 cells, but with millions or trillions of cells. These organisms are not transparent, take a long time to develop, and consequently, different approaches have been utilized in order to try to map cell fate, including the very elegant studies of Nicole Ledorraine, who performed transplants of quail to make quail-chicken hybrids, but one can imagine that that can only go so far. Then there are the very elegant spectrally distinguishable fluorescent proteins epitomized by the development of Brainbow. But an important point to make about that technique is that it clonally marks populations of cells. And clonality can only get you so far. At the simplest level, we are all clones of a zygote. If you look at a tumor, that's frequently expressed and emphasized that it has clonal origins in a single cell, yet we now know there's tremendous subclonal heterogeneity. And so if you have as few as 4 cells, there are actually 15 different cell lineage histories. And if you double that number to 8, there are 135,000 different cell histories, and go up to 16, and it soon approaches Avogadro's number. And the point is that within a clone, there's a tremendous amount of precise lineage information that can't be resolved. So what one needs is a virtually limitless supply of markers. Steve Salaponte and our group first addressed the possibility of using sporadically arising somatic mutations in order to resolve lineage. And this works, but it works to a certain extent. Michael had very nicely summarized some of the limitations of the approach, that the mutations are hard to find. However, you can identify them using brute force approaches, as Michael Stratton and colleagues did when they sequenced the whole genome of many individual cells that have been amplified from a mouse through embryoid body formation, and were able to construct precise deterministic lineages. However, due to the limited number of mutations that were identified, the cell trees could only go so deep. With that in mind, Jay is going to tell you about a new engineered approach that has turned out to work incredibly well.
Okay, so I had this slide in here. This was actually my first project in graduate school. Was I think inspired by some of the work that Chris Walsh had done actually when he was a postdoc with Connie Sepko and some other work going on at Harvard at the time. I was thinking about this idea of how to multiplex lineage tracing. And so this was maybe 15 or 16 years ago. Came up with ideas that I think echoed some of the things that Michael and Long talked about with their terrific system, and also similar to the Brainbow system. And I worked on this for 6 months, threw up my arms in failure, and went on to work on some other things. But kind of had this in the back of my head. And actually, even in starting my lab, I didn't really pick back up on it. I knew that Marshall had been working in similar areas, and it wasn't actually until I think I got the email from Kathy about this that Marshall and I reconnected about this, and I started working on this again with him. So, okay. So the system, I think you'll see some similarities with memoir that Michael presented, but also some, I think, complementary differences. Is the idea of using CRISPR-Cas9 as a means of recording lineage information. And so there's a lot of advantages here. I think the focus or the system that we came up with involved trying to pack as much information content as we could into a compact space. And so this was work that was led by 2 graduate students, Aaron McKenna and Greg Finley. Okay, so I'll just walk through how this works. So our acronym is GESTALT. I like memoir a little better. It's a little more fitting to the method. But so we have got a barcode here that consists of about 10 target sites for guide RNAs. And the idea is that as development progresses, you have accumulation of point mutations, but more often insertion and deletions through non-homologous end joining, but different events in different cells, meaning different precise mutations. Even if a given site is edited, it can be edited in many different ways. And then that these accumulate irreversibly and combinatorially into this relatively compact space that we can then, in principle, read out by sequencing. And then the lineage information is recovered from the sharing of edits between cells. So we did do some cell culture work prior to this. I'm just going to skip to the animal experiment where we tried to take what we thought was a working system in cell culture and put it into a fish. And so for this we teamed up with Alex Shear's group, and in particular a terrific postdoc, Jimmy Gagnon, there, who led a lot of the fish work here. So one of the advantages here of trying it in fish is that we could try it very quickly. So we have this barcode that we put in to adult zebrafish as a transgenic, and then we can inject all of the editing reagents, including guides corresponding to each of these 10 targets, and Cas9 into single-cell embryos. And then we just let development proceed through embryogenesis and then onward to adult stages. So while we were waiting for the adults to grow up, we could take a bunch of these embryos and look at them to see if this was working. And so one thing that we see here is that we do see a lot of alleles. So when we sequence over the barcode region, this is just PCRing and sequencing that barcode, we see diversity accumulate. So even though we have only 10 target sites, we're accumulating thousands of unique alleles. And if we compare different embryos to each other, we don't see sharing of alleles, which convinces us that when we see the same allele twice in a given embryo, it's truly by descent rather than by chance. The other thing we noticed here, so every group here corresponds to a developmental time point going across here, and every bar is a different embryo. And so even though we see more alleles later in time because we're sampling more cells, most of the editing happens relatively early. So we're not encoding the editing reagents genomically, and so you can imagine they just crap out after a certain number of cell divisions rather than continuing to edit. Okay, so this is what the semi-partially processed data looks like. So every line here represents a unique allele, and the histogram over here is showing how many cells we're seeing that allele in. And we primarily focus on insertions and deletions because of the bioinformatic challenges with these kind of data. And so the blue bars are insertions, the reds are deletions, and as you can see, there's quite a diversity. But we also see that certain editing events are shared between cells. So for example, almost exactly half of the cells in this particular embryo have an insertion at this first site, which is consistent with an editing event that happened at the 2-cell stage. Right? Okay, so the informatics here are challenging. So most software for kind of phylogenetic inference in the genomic space was developed around point mutations that occur in evolutionary time, and here we're dealing with a very different matrix of likelihoods for different kinds of mutations. But we were able to come up with software for inferring phylogenetic trees based on the alleles that we observe, right? So here, for example, this is a clade, and you can see that every member of this clade is defined by or shares a common pair of edits, right? And therefore, we can group them together. And then you have subgroups that have dependent edits that are only present in subsets of the alleles. So, okay, so this is one portion of the overall tree. So if we zoom out, we have these. So this just keeps wrapping over, but we have these Selston-like trees where we have over 1,000 alleles that we're able to pull together into this single tree. And this was from a 30-hour embryo. And I think at most we're getting probably 8 or 9 levels in terms of the depth of the tree that we have. Okay, so it seemed to be working. So we waited a few months, got our adult zebrafish, and so in the embryo experiments we're just sampling cells. We don't know what cells are who. We do a little bit better here. We're able to separately sample different organs from the fish and look at the alleles that contribute to them. So remember, all of the editing happened early, so we're really looking at those 1,300 or so alleles and kind of what contributions they're making to different organs. And so we did a more focused analysis on the heart, where we took off a chunk. We also sorted out cardiomyocytes and non-cardiomyocytes to look in a little more detail. So one nice thing here, when you're using sequencing, you're able to sample an incredibly large number of cells. So here we're able to look at nearly 200,000 cells from diverse organs, but these boil down to around 1,000 alleles, right? So we're seeing each allele in many cells. Okay, so one surprising finding, and we've seen this again, the numbers and the proportions are obviously not always the same, but we've seen this in a few different fish, is that only a handful of alleles contribute to blood, right? So one thing I didn't know before getting into this is that zebrafish red blood cells are nucleated. And first we just thought we had some weird contamination of these 5 alleles in every tissue, then we realized it was just blood. So if you sort out the blood and look at it separately, we see these 5 alleles that account for almost all of blood. And then if we look at each organ, those same alleles are present at kind of the same ratios relative to each other, right? So they're there, quote, contaminating, except for the sorted cardiomyocytes, which obviously don't have blood. And so when we look more broadly at organ systems, we see a similar trend. So this is a histogram of the fraction of cells that are explained by the most dominant alleles, where the alleles correspond to these marked early progenitors. And so the blood is the histogram in red, where you can see almost all of it's explained by a few. But for every adult tissue that we looked at, 6 or fewer barcodes account for more than half of the organ, right? And you can ask, like, well, are these the same alleles that are present in different organs? Or are they sharing across different organs? And for the most part, the finding is that the alleles that are prevalent in any given organ are not prevalent in other organs, right? And there are some exceptions to that. So maybe we see some sharing between different parts of the intestine. And also, if we're looking at the heart in greater detail, so that's one barcode we see in the heart, but it's not really prevalent in any other organ. And this big block of alleles that are related to each other are all different, all come from the heart, right? So that may explain that sharing. So just as a hat tip to our afternoon experience yesterday, when we were putting together this paper, we initially tried to actually represent this as Mondrian type art. And so Jamie Gagnon, the postdoc I mentioned, loved this, as did I. And so we made these nice pictures so you can see so the diversity in terms of the embryo versus the adult, and then looking in specific organs like blood, which has very little diversity, or even brain, which doesn't have so much. A lot of alleles, but a few alleles explaining most of it. But we were outvoted by our colleagues, and this didn't end up making it. Okay, so this is the overall lineage of the adult. Just over 1,000 alleles in across the whole organism here. I'm going to speed up a little bit here for time. So if you go back a slide here, so you can see that if you look in a little more detail, you can see that there are kind of these major clades, right? So big groups, about 8 big groups of cells that we can relate to each other on the basis of what were likely very early edits in the embryo. And so here I'm just zooming in on one of those. And then we can look at, if we look at the organs in which those alleles appear, it looks a bit all over the place, but we do see non-uniformity in terms of progenitors defined by all the descendants of this guy here and how they contribute to different organs. So for example, this is looking at these 8 inferred progenitors, and we can see that some of them contribute to many organ systems, but others, even though we may have thousands of cells represented, only contribute to neuroectoderm or only contribute to mesoderm. So we can look at this in a more nuanced way, going from an ancestral edit to successive additions of more edits on top of that, and we can kind of watch or visualize increasing amounts of restriction in terms of the organs in which we see those alleles or groups of alleles appearing in. Okay, so what's next? So we've been trying to, one great thing about CRISPR-based systems is that you can, in principle, port them to any model organism that CRISPR works in, which is, thus far, we haven't really seen the limit of. I think one major limitation of the way that we've done this, and the difference from, let's say, a Sulston-like map, is that we don't, other than a very gross picture of anatomy, we're lacking information as to what each of these cells are, right? And also where they are, right? So spatially where they are and functionally who they are. So I think Michael and Long's work really nicely addresses the spatial piece. We've been trying to focus on functional identity to try and get more out of this lineage information by also trying to concurrently capture functional information on each cell that we're sampling. And so our lab, and I know many others, have been working on single cell techniques. We've been trying to develop an atlas of chromatin state in the mouse. And so this is based on a single cell ATAC-seq protocol that we developed, and we're over 100,000 cells now. I should emphasize this is not a major production thing. This is work from a postdoc and a technician over maybe 2 months. And we can do this because we rely on this kind of split pool method that is high throughput. And so we can take these data sets, we can break them into clusters, and compare them to single-cell RNA-seq and see similar kinds of clusters that we can map back. But obviously, we'd like to have lineage information on top of this. And so one of the graduate students had the foresight to put the barcode under expression. So we have it in RNA, and in principle can recover it in single-cell RNA-seq data sets. So we've just started to do this. This results just from the last maybe 72 hours or something like that. So this is a single-cell RNA-seq data set of about 600 cells from one of the GESTALT zebrafish embryos. And so you see these clusters. We can break these down into different cell types based on markers that are overrepresented. And then in a proportion of these cells, we're also getting the lineage barcode. And so we can also reconstruct a GESTALT-like history and then map what cells we're seeing over here to the lineage information. And so we don't have a great way to visualize this yet, but at least for blood, we see something very consistent, which is the blood that we're seeing here in this is an embryo, or blood-like cells in the embryo. Remember, we didn't dissect this. Correspond to just a handful of barcodes in the lineage tree. Okay, so I'll stop there. I'm probably over. So again, I think that where this is headed is really an integration. I think we want lineage histories, but we also want them to be coupled to, I think, not just molecular profiling data sets, but also spatial information to really kind of put the whole story together. And I'll stop there. I'll just add my thanks to Marshall. This has been one of the funnest meetings I've attended, or probably the funnest in memory. And thank you to Tom and Kathy and Nicole for organizing. It's been really terrific. So.
Questions?
Michael.
Yeah, really beautiful talk and great system. And I'm just wondering whether there's any correlation between the diversity of barcodes that you get and the stage of development at which a particular organ starts to expand or proliferate.
So one interpretation of the data is that certain cells take over early. Another interpretation is that there's late remodeling of organs and sort of late stage dominance. I don't think we know yet. I think we have to go through and do more of a time series to really get a handle on that. The one precedent for this that I think encouraged us to believe the result was real is that there's work in the heart, zebrafish heart, that shows that there's late stage remodeling where certain clones take over, right? And so that might be more generally be the case, right? Chris.
So that's really beautiful work. Did you say there's most tissues have contributions from like about 8 major clades or something like that?
I said that most. Oh, you mean how many? Most tissues have contributions. The majority of most tissues, or all tissues, was 6 or 7 alleles. If you look at the clades, I would say most of the clades are actually contributing to most of the organs. There are a handful of clades that only appear to contribute to, let's say, ectoderm or mesoderm or so on and so forth.
Okay. How are you distinguishing alleles from clades?
So clades.
You mean alleles meaning?
Alleles meaning the final allele.
Thing, and then the clade
meaning the inferred progenitor, a group of alleles that all share some early edit.
Okay, great.
Right.
I guess, and then the last question is, assuming that
the alleles are very tissue restricted, the clades are not so much, but you can see some restriction.
Okay. So assuming that humans are similar to zebrafish in terms of the early differentiation of blood from other tissues, how good or bad do you think blood is as a proxy for, say, somatic mutations in other tissues? Is it good for some tissues and not others? Is it bad for all, or what do you think? What's your general sense?
That is a great question. I think it's worth actually a focused analysis to try and assess that based on the data. Obviously, doing it in mouse would yield a more relevant result. But in principle, one could do that analysis directly from this kind of data. We just haven't done it. But my gut feeling is that for early events, you would see them in blood. Actually, well, I'm not so sure, actually. I'm not so sure.
I would just add that people, there's a lot of selective pressures that accumulate with age. And so a huge area of investigation in hematology is clonal hematopoiesis of indeterminate potential. But that is, a pre-neoplastic phenomenon that's well beyond looking at developmental origins.
If you were to look back, I think maybe only 2 of the clades contributed to blood. Right? So only 2 of only 2, because some of those 5 alleles were actually related to each other. So.